question. You've got the genome to the axolotl, you've got the genome to the frog. How come we don't have any models that tell us whether frogolotls will have legs? We have no idea from that information whether frogolotls are going to have legs or not. And if they do have legs, whether those legs will be made of axolotl cells or also include frog cells. We have no idea, even though you've got the genetic information. So it's really important to start to understand the algorithms, because where biomedicine is right now is that we're very good at manipulating molecules and cells and getting information like this, which gene and protein talk to which other gene and protein. We are a long way away from actually controlling a large-scale form and function. And in fact, you can think about the kind of parallel journey that computer science took. This is what programming looked like in the 40s and 50s, where in order to control the system, you had to physically rewire it, right? You were pulling wires in and out, you had to rewire it. Nowadays, for a joke, I say to all my students, you know, you're on your laptop, you're going to go from Microsoft Word to Photoshop. I want you to get out your soldering iron and start rewiring. And of course, they all laugh because nowadays we don't need to do that. We understand that if your hardware is good enough, it's reprogrammable with stimuli, with inputs, with experiences, not rewiring. But of course, modern molecular medicine is all about the hardware. We're all very excited about genomic editing and protein pathways and single molecule approaches. And so I think the reason that we are still roughly where computer science was in the 50s is because we've been neglecting one important aspect, and that is multi-scale intelligence in biology. Now, what do I mean by intelligence? I mean what William James meant, which is the ability to reach the same goal by different means. And when we talk about pattern and anatomy, when I talk about goals, I mean regions of morphospace. Now, what's morphospace? Morphospace is the space of all possible configurations of some particular structure. So if you're looking at snail shells, for example, there are three till they get to where they're going. Parenthetically, this is not just for frogs and for axolotls and worms. The human liver, of course, is regenerative. Even the ancient Greeks knew that. I have no idea how they knew that, but it seems like they did. Deer every year regenerate massive amounts of bone, up to 1.5 centimeters of new bone per day. Bone, vasculature, innervation, skin. And even human children below a certain age can regenerate their fingertips. If you just leave it alone, it'll basically regrow cosmetically. A very, very nice outcome. So we have some ability to improve our position in morphospace. One of the most amazing things about it is that as a body, as a living creature, you can't count on your environment. You can't count on, in other words, you can't count on the environment being the same as it was before. You can't count on not being perturbed during the developmental process, maybe physiologically, maybe metabolically, maybe with a parasite or a teratogen. So you can't count on that, and you still need to get your job done. In fact, you can't even count on your own parts being what you expected them to be. Now, of course, we don't have any machines that can do this, that can repair themselves after damage or put themselves together with diverse parts. Here, this is one of my favorite examples of all time. This is a cross section through a newt kidney tubule. So here's the lumen of the tubule, and these are the cells that make up the tubule. Normally, it's let's say around 8 cells that work together to form this kind of tubule. But one thing you can do is you can make these cells what's called polyploid, which means they have extra genetic material. Amazing thing number one. With the excess genetic material, you still get a perfectly normal newt, no problem. That there are all kinds of extra information around, no problem. But what does happen is that the cells get much bigger. And as the cells get physically bigger, fewer of them are needed to make the exact same shape lumen. Now that's amazing thing number two, is that these cells, the number of these cells will actually scale to their correct size so that you get the same final outcome with different numbers of cells. But the most amazing thing is that when you make these cells absolutely gigantic, so that only one cell is big enough to make the whole lumen, what it will do is it will no longer cooperate with other cells. One cell will bend around itself, making the exact same lumen. Okay. Now, the incredible thing about that is this is a different molecular mechanism. This was cell-to-cell communication. This is cytoskeletal bending. And so what happens is that this is a kind of top-down top-down causation, where in service of a large-scale anatomical spec, meaning having the correct lumen, you can call up different molecular mechanisms to get the job done. So this, again, is sticking with this theme of intelligence as the ability to handle novelty in terms of getting to where you're going from diverse starting positions, with perturbations, both external and internal. You know, your own parts are changing. Can you still do what you need to do? For moving the body configuration in morphospace. Before there were nerves and muscles, cells needed to talk to each other and process information to get around in morphospace. And so we've argued in this paper that basically neural electricity is just an evolutionary pivot across spaces from morphospace to three-dimensional space of a very system for processing information long before brains arose. So in my group, we were thinking about this, and we asked this question: okay, if that's how it works, could we make some tools to read and write this electrical information and to really understand how it's navigating that shape change space? So we developed some of the first tools to read and write information into non-brain tissues. So of course these dyes, so these are voltage sensitive fluorescent dyes that report all the electrical conversations that these cells are having with each other. So here's a time lapse of these cells figuring out who's going to be left, right, dorsal, ventral. So all of this is you're looking at these cells having those conversations. We do a lot of computational modeling to ask, okay, given where the ions are going, how can we predict these voltages? I'm going to show you two patterns. Here's a time lapse. It happens to be grayscale, but it's the exact same idea. Here's a time lapse of a frog embryo putting its face together. And what you will see, and this is one frame out of that video, what you will see is that this is something we call the electric face, discovered by my colleague Danny Adams. When she was in my group doing this profiling, she found that here's a particular frame from that video where you can see the animal's right eye, the mouth, the placodes. All of this is already demarcated before the genes start to come on and really pattern the face. This is a bioelectric pre-pattern of what the future face is going to look like. And I'm showing you this one because it's so simple. It literally looks like a face, right? There are other patterns that are much more complex that you can't really just, sort of visually decode. But this one is very clear. This is the memory of what a tadpole face is going to look like. And if you move this, if you disrupt this electrical pattern, the gene expression will change and the organogenesis will change. And I'll show you examples of that momentarily. So this is an endogenous pattern that is required for normal development. In fact, human channelopathies human patients that have craniofacial defects often have mutations in ion channels that screw up this pattern. Now that's a normal pattern. Here's a pathological pattern. We've put in a human oncogene. These cells will make a tumor, but even before the tumor becomes apparent, you can already see this aberrant bioelectrical signature of these cells basically disconnecting from the rest of the tissue and going to their unicellular ancient behavior mode, where the rest of the body is just environment as far as they're concerned. So one set of tools is to track these bioelectrical changes. Now here's another important set of tools to actually start to be able to write new information into it. And the way we do that, we don't use any applied electric fields. There are no waves, there are no magnets, there's no electromagnetic radiation. What we do is we do exactly what neuroscientists do, which is we modify the endogenous mechanisms by which cells establish electrical signals. So either ion channels, we can open them, we can close them, we can use light or drugs to do that. And the gap junctions, the electrical synapses, so we can determine which cells talk to which other cells by opening and closing these little gap junctions. It's very telling that all of the tools of neuroscience work in other cell types. Basically, the tools can't tell the difference, right? This distinction between neuroscience and other cells in your body is completely artificial, and it's a consequence of humans trying to parse the world. It's not. Nature doesn't obey that distinction. So what can you do with this? Okay, I've been talking about this electrical information. What does it actually do? Well, here are a few examples. One thing you can do is you can take this tadpole, and I've shown you the electric face. And so we asked the question, okay, if now that we know that there's a particular voltage state that kickstarts eye development, could we simply reproduce that somewhere else? So we took an ion channel RNA that we knew was going to set the same voltage state. We injected it into precursor cells that are going to give rise to the gut. And sure enough, those cells can form a complete eye. The eyes that are formed will have all the right layers, retina, nerve, optic nerve, all that, lens, And they can be formed anywhere. They can be formed anywhere in the body, as long as the cells get the right pattern that tells them what organ to make. Now, notice two very interesting things here. One is that this instruction is highly modular. We provided a very small piece of information, just a voltage pattern. We certainly did not give it all the information needed to specify an eye. Eyes have many cell types. They're very complex. We have no idea how to micromanage the creation of an eye. But what we provided was a kind of a subroutine call, a signal that says build an eye here, and the cells do. That's the first thing. The second amazing thing is that there are two levels of instruction here. One is us instructing the cells build an eye, but here what you can see is this is a lens sitting out in the flank of a tadpole somewhere, and the blue cells are the ones that carry this ion channel that we put in. But there's not enough of them to make a good lens. So what they've done is recruit their neighbors. This is why at the very beginning I pitched this idea that frogolotls might have legs that consist also of frog cells. Why would frog cells build a leg when they normally don't? It's because cells have the ability to instruct each other about what to build. And so these cells right here, they've been told to make an eye, but there's not enough of them, and they know that. And so they recruit a bunch of their neighbors, these brown cells that we never touched. They're not, these cells are completely wild type, and yet they're participating in making this ectopic lens out in the tail of a tadpole. So there's this amazing ability to instruct, and for the cells to then instruct each other. Now, what else can we